Hello, BIO178. In this video, I will cover some of the logistics for figuring out how to write your approach section. Now, I wish there was a simple formula I could give you where I say do X, Y, and Z, and then you'll have your approach section. Unfortunately, it's not quite that easy because all of you have different proposals and slightly different experiments, which is great. That's what I want to happen, is for you to all focus on things that are interesting, especially for you, but that also means there isn't really a one-size-fits-all type of way to do this. So what I'm going to do instead in this video is walk us through some guiding questions to guide you in things you should be thinking about, to think about what information to include in your approach section. This is also going to be a graded survey for this week where you're going to have to answer these questions again to help you build toward that approach section. So again, how do you figure out what approach to take for your proposal? I used this picture before, but I think it still applies. It can be intimidating when there's all this literature out there and it might be hard to figure out where to start. The good news is, at this point, you have already put a lot of thought into your proposal in terms of coming up with your research idea and your aims, so I think you have a pretty good place to start. But I'll try to provide some concrete questions and things for you to think about to help you come up with that approach section for your proposal. So the first thing that I recommend is finding a or multiple reference papers that investigate a similar study question as the one you are proposing. So again, you may already have a few of these papers that you've been reading for reference as you've been working on your proposal so far. So for instance, if you were interested in doing a study looking for molecular evidence for ecological divergence or local adaptation, this might be a good paper for you to reference. And then specifically what you should do is focus on the method section of that paper and think about what approach did those authors use to answer their question, and you could use a very similar approach, similar type of experimental design for your study. One thing I will mention is that depending on what your study is and what kind of papers you find, you might need to kind of mix and match a couple different papers to use for reference. So you might need one paper, for instance, that gives you some information about the sampling methods for your particular organism. And then you might need another paper for the molecular methods or analysis. So for instance, if you were interested in looking at genomic evidence for local adaptation, you could use this paper for an example of the molecular methods. But say you were proposing to do this in some large mammal like mountain lions or bears or something. This paper wouldn't be informative in terms of collection and animal maintenance. This paper talks about how to maintain and collect snails. That's not applicable. So you could use this paper for a molecular methods reference, and then you could find another paper that describes for your particular study organism how you might go about sampling from that individual to get that molecular sample that you need. Next, what I'd recommend is considering the general experimental design. So again, I showed this picture in the last video. So when we're thinking about our general experimental design, what we want to think about is, are we looking at a single population or are we looking at multiple populations? And in either case, make sure you're thinking about which populations you're studying and what your sample size should be for each population. And then think about, based on whatever you're proposing, do your organisms need to be brought back into the lab? If you're doing some kind of local adaptation, common garden approach, you might need to collect individuals from the field, bring them back into the lab. If so, what type of conditions are you going to keep them in the lab? All of these are things for you to think about. Next, you want to think about what type of sample you're going to obtain. So for each individual you've just decided you're going to collect, what kind of sample are you going to actually get? Is this going to be a tissue sample, a hair sample, blood sample, feces sample? How are you going to collect those samples? So again, this image on the left is two of my previous undergraduate students collecting tissue samples from the snails. You might need to get some type of tissue sample. Or in this picture on the right, maybe you need to collect feces. Maybe you're collecting water samples if you're doing an eDNA approach. If you are doing a study where the timing of it is sensitive, you also need to think about what time of year you're going to collect your samples. So if you're looking at a gene expression study, for instance, gene expression varies throughout the year based on different seasons, so you want to make sure you're taking that into account. Same kind of thing with an eDNA study here on the right. You want to make sure you're taking into account what time of the year you're actually taking your samples from, because that can affect your results. And then we want to think about what molecular method you're going to use once you actually collect those samples. So are you going to use something like a metabarcoding approach, which is shown here? Or are you going to do some type of high throughput sequencing, like maybe whole genome sequencing? So think about what type of molecular method that we've already talked about so far in class are you going to use? And then what you want to do is, now that you've thought about all of those things, what you want to do is connect your approach to your research question. So once the molecular methods you've described above, so say you chose a metabarcoding or whole genome sequencing approach, what data are you going to get that's going to allow you to actually answer your research question? So make sure you have that connection between what you've done so far, the approach that you're taking, and again, how it's actually going to answer your research question or accomplish your specific aims. So an example of this, it could be something like outlier SNPs. So maybe you're going to identify outlier SNPs, and that's the data that you're going to get to allow you to answer your research question. This image on the right we've shown a few times. Each one of these blue dots in the red region is an outlier SNP. Or maybe you're taking more of a species ID approach where you're trying to identify what type of organisms are in the microbiome, for instance. Then you might do an approach like we did in our lab a few weeks ago. We were looking at the microbiome of the male versus female snakes, garter snakes. So you might have an approach like that, which is shown on the left. And then once you've figured out what type of data you need to allow you to answer your research question, you also want to think about the analysis programs that are going to help you obtain that data. So in this case, when we did this metabarcoding species ID approach, we used DNA Subway, but really that was just an interface for this program called KIME. Hold on, I can't remember if there are two I's. Yes. 2 I's, 1 M. Okay, KIME is a program that DNA Subway actually uses to do this species ID. Or if you're doing outlier SNPs, there are certain RStudio packages you can use to accomplish that. We're actually going to use one of those in lab very shortly. So think about the data you're going to get and then what type of program you're going to use to analyze it. So as you're thinking about putting all of this together in terms of roughly thinking about how long this section should be, your approach section overall should be about 250 to 400 words. Again, we don't have a lot of space in 2 pages. And remember that we have 2 different aims, so you need some kind of approach description for each of those aims. In this example here from my GRFP proposal, these 2 sections here I've just outlined are the approach. This is the approach for aim 1, and this is the approach for aim 2. So the guideline I have up here, 250 to 400 words, is for the entire thing. So if you think about that, breaking it up in 2, aim 1 should be like 130 to 200 words, and same thing for aim 2. All right. So that example that I just showed on the previous slide that you couldn't read at all because it's way too small, I do actually have that posted on Canvas so you can see a full example from my proposal focusing on fish eggs, which is that image that's shown here. I also have another example for us, and I've created a Canvas quiz where what we'll do is I'll give you the approach section for this other funded GRFP proposal. This is from Dr. Caitlin Louder, who was in grad school around the same time as me, and she got a funded GRFP to look at spiny lobster, which are a common species in La Jolla in Southern California. So what I'll do is for this Canvas quiz, I'll give you that example approach section, and then we'll have you pick out the different sentences that she included to reach and address each of the different, that's the word I'm looking for, the different criteria that we just talked about in these videos. And so just to recap and think about where we are and how far we've come, in our first proposal one, you included a title, introduction, research idea, and 2 specific aims, along with references. Now we're fleshing out the rest of that proposal. So what we're doing here is adding in that approach section to provide details about how we're actually going to accomplish those aims. And then we'll keep building on that. And very shortly, we'll get to the point where you'll have all the information you need to write a full NSF GRFP research proposal. So keep up the hard work. I know proposal writing can be intimidating and you can go down rabbit holes when you start reading all these different papers, but keep up the hard work. I know that you all can do it, and I know you're doing a great job, so keep it up.